Yes, sir, you're live now. Please.
All right, very good morning, everybody, and all the MOOC students and participants. Welcome to the third live session, which is last live session for this course. Today, we are going to talk about protein microarrays and label-free biosensors, which is the key theme of the Interactomics course. And rather than giving you another lecture about all this technology, which you have been already studying in the course, today the goal is to give you the more practical hands-on activities and the live session from the lab to acquaint you with what all goes into the doing the research in this area, right? So let me first start with the just refreshing you on the concepts of protein microarray, which you have already studied in quite elaborate manner, right? So there are different type of microarray platform which you have been studying. For example, one could take the antibodies, print them in different ways on the array surface, and based on that, either you can do the direct labeling, or you can do the sandwich assay, or you can take the tissue blots and print those and use antibodies to look for the abundance. That is the reverse field arrays. Conventionally, people like to purify the proteins and print those on the arrays. Those are known as the protein arrays, which is essentially with the target proteins, chemical linked. Or if you are using the mass spectrometry to identify the peptides, and you know which peptides are the best sequences, you can even synthesize those peptides, synthetic peptide, make those peptide fusion and print those on the chip, the peptide arrays. Or you have also learned about different type of cell-free expression-based system. For example, you can take the DNA and you can do the transcription and translation on the chip itself, right? That is with the cell-free expression-based methods. And NAPA, which we have talked in quite detail, nucleic acid programmable protein arrays, they are one of the strong platform for doing the cell-free expression-based arrays, where you are starting from the a gene of interest, which is having the GST tag, and that you are going to print on the chip. You are going to add the machinery to do the in vitro transcription translation, and then based on that, the protein gets synthesized. That protein contains the GST tag, and on the array, you're also keeping the capture antibody anti-GST, so that the expressed protein which contains the GST tag, it's bound to the antibody, capture antibody. Or you can use any pair of antibody for that matter, like the flag expressing in the protein and the anti-flag on the chip, and they are binding together, right? So these are the different ways of doing the arrays. And finally, people have also tried to take the unpurified PCR products, printed them on the chip, and then added the in vitro transcription translation machinery on top of it, which is known as the multiple spotting technique or MIST. So these are just kind of broad bird's eye view of the different microarray platform which you have been studying in the course. These protein microarrays have many applications which have a lot of clinical implications. Often, like you've also been exposed to some of the mass spectrometry based technologies, and which also appear to be quite robust, right? But when it comes to looking at the biological sample, the biofluids, blood sample, plasma sample, cerebrospinal fluid sample, other biofluids, therefore, the protein microarray actually gets edge over the mass spectrometry for analyzing, uh, because here we are not doing any kind of pre-processing of the sample to remove the abundant protein. For example, in the plasma or serum sample, you have a lot of abundant protein, albumin, immunoglobulins, which for the mass spectrometry based approaches, you need to deplete out and remove those abundant protein so that the low abundant protein get expressed. In case of protein microarray, our readout is different. Our readout is with the fluorescence, which can go 10 to the power 10, whereas in the mass spectrometry, dynamic range is very narrow. So you need to bring all the protein in that range. Therefore, protein microarray has got more application for the clinical samples. And therefore, for the biomarker discovery or looking at some of the novel targets, people prefer protein microarray as one of the platform. Also, the amount of sample which you're going to probe on the chip, these are, you have seen microscopic glass slides for your different bio courses. On these chip, you can take few microliters of your biological sample, dilute with the buffer, and probe on the chip which contains your antibodies or proteins, and then place a cover slip on that. So now with a very small amount, still you are able to probe the thousands of protein on the array. Whereas in the mass spectrometry, you need quite a bit of amount of sample which you need to deplete out and extract the protein and then run on the mass spec. So therefore, I'm just trying to give you the contrast and looking at the different technologies and where some technologies can be more beneficial. So for example, again, microarrays are currently not as heavily used as the mass spectrometers, but they have very unique and niche area which mass spectrometry cannot replace. Again, if the goal was to look at a lot of functional proteins and look at the functionalities of these protein, unknown proteins, where they function, what kind of interacting partners they have, for all of this, protein microarrays are the very beautiful, elegant platform. So as I said, one application which protein microarray gets the edge is looking at the markers, biomarkers for different diseases, especially looking at autoantibody-based biomarkers. In this case, some of the patient's serum samples can be taken, probe on the chip. If towards a given disease some antibodies are produced, they will bind with the antigen on the chip. And now, looking at the secondary antibody responses, you can actually look at which those targets are of interest. And if there is some pattern emerging across multiple patients, then those could be potential biomarkers. Ideally, one is also looking at protein arrays approach to look at protein interactors, the proteins bind to which other protein. And here you can look at the very unbiased high throughput approach. One could also look at the different type of modifications, PTMs, right? So again, protein microarrays has a lot of applications. And while my lab does protein microarray for many clinical problems, but today what we thought on a thematic side, we will take one case study on SARS-CoV-2, how we utilized protein microarray and peptide microarray platform during the COVID-19 to do research on the SARS-CoV-2. In fact, we're still continuing research in this area on using different type of array platforms. So to do this, idea was to look at the entire SARS-CoV-2 proteome, and which is not very large. So for each of the protein, almost 15 amino acid long peptides were synthesized synthetically. An idea was if we can have the overlapping fragments, right? So with these overlapping peptides, one could actually, like some residues will be common, right, across different peptides. But then these overlapping peptides could be placed and printed in a way that they can represent the full length of the SARS-CoV-2 proteome. And based on this, now with the single chip, one could screen for all the proteins of SARS-CoV-2 and use that in different ways of looking at epitope mapping or antibody biomarker identification, looking at humoral immune responses. Many applications can be tried out on this area. So how to do this experiment, right? Let me now show this particular video which we have made from our lab, how we perform these experiments. And that will give you an idea about step-wise how these experiments are being performed. So let me just play this thing for you.
Today, we will be demonstrating peptide microarray technology. The first step involves reagent preparation, namely washing buffer, staining buffer, and Rockland blocking buffer. After preparing the reagent, we move on to incubation tray assembly. Unscrew the incubation tray by slowly turning the screws diagonally. We will be using two blank glass slides as balancers, since today we will be using only one microarray chip. This is the pepperprint microarray chip. Slowly take out the chip using plastic forceps and place it on the incubation tray. Align the chips properly onto the tray. Now, allow the gasket to be placed over the slides. Remember to keep the glossy side of the gasket on the downward side. Align the silicon gasket over the slide and tightly seal the incubation chamber using the screw. Remember, tighten the screws only diagonally. This will ensure that the slides do not break. Next, add 2 ml of staining buffer onto the chips. This step is called equilibration of peptide microarray chip. Please note that we are not touching the microarray chip at any point. Now allow the equilibration process to happen for 15 minutes. Now aspirate and remove the buffer and add 2 ml of blocking buffer. Slowly add the buffer onto the slides. Allow for 30 minutes of incubation. The next step is sample incubation. To the staining buffer, we will add patient plasma at a dilution of 1:200. This is the patient plasma. Now slowly overlay the diluted plasma over the slide and incubate overnight. This will be done in a cold room at 4 degrees Celsius at 140 RPM on an orbital shaker. On the next day, remove the incubation tray from 4 degrees Celsius and aspirate the solution. Now add washing buffer thrice onto the chip. This step is done 3 times in the dark. Again, completely aspirate the washing buffer and add secondary antibody. Prepare of dilution of secondary antibody 1:2000 and then slowly overlay the secondary antibody on the microarray chip. For 45 minutes, allow the incubation to happen at 140 RPM at room temperature. Make sure that you do not expose the slides to light at any given moment. Now remove the buffer and wash the slide thrice using washing buffer. Now gently take out the slide and put it in dipping buffer. This is done to wash away all the salts that may be over the peptide chip. Use a pressurized stream of nitrogen to completely dry the slides. Finally, we will scan the slide. This is the microarray scanner, GenePix 4000B. We are using manufacturer recommended settings for scanning the slide.
All right. So what you got a glimpse of the entire microarray workflow. which is essentially if even at the your molecular biology labs, if you have done some basic experiments on Western blot, right? Uh, it is very similar approach here. The way you do in Western blot, you are going to block the slide, your membrane. You're going to probe with the primary antibody, then secondary antibody, and then you want to develop the reaction and scan that, right? Similar way, we are also doing microarrays. It's very similar concept. Only difference here is we are looking at the samples directly, right? And that we are going to utilize often as the primary antibody source. So I hope this will give you the more relief. That is not something which is very difficult technology to work with. Even if you have done the simple SDS page or Western blot, you should be in a position to do these experiments in your lab. But now, after scanning the slide, this experiment was 1.5 day take because of incubation and overnight incubation steps and then washing step. But then you got high density data from the slide, which is having thousands of feature printed, depending on what kind of slide we are using. In the SARS-CoV-2, of course, you have less number of features. When we talk about human proteome arrays, we have, several thousand, maybe 40, 50,000 spots printed. So it gives you the big data, right? Just after one scanning of the slide, what you get in Excel sheet, lot of intensity value for each of the spot, duplicate different type of blanks, controls, and then how to make sense of the data. That becomes quite challenging. And that's, I think, as a student, it is good learning for you to understand how to make sense of that data. So now I have invited one of our senior PhD student, Arka Ray, to talk to you about data analysis. He comes from the electrical engineering background and also he's developing his own indigenous microarray device, but has been quite instrumental in looking at data in different ways and different type of programming to be implemented to make it very streamlined. Of course, he has a lot to talk, but maybe in a short, he will give you a snippet of the different steps involved in doing the data analysis. So, Arka, please share your slide and turn on a video, and you can join for data analysis session now.
Thank you, sir. Good morning, everyone. Now I'm going to show a few steps of microarray data analysis. Majorly, will focus in the protein arrays and SARS-CoV-2 that viruses and how we are using protein arrays for that. So let me share my screen. I hope my screen is visible.
Yes, go ahead. Full screen. Go ahead.
Yeah. So initially, we need to for the data analysis side, few data extraction also a very crucial part. So for that, we require a few softwares. So how do you extracting the data? Then followed by that, we go ahead for the data analysis. So now there are different instruments we are using in lab. So for our lab, we are using GenePix for scanning the slides. Then followed by for already sir has mentioned regarding protein and peptide arrays. For peptide arrays, we use different software for the data extraction that is called Mapix. But for protein arrays, we dedicated for the data extraction side, we go ahead with the GenePix because the scanner is GenePix, and finally the software we use for the analysis for the data extraction, that is GenePix Pro. For the initial scanning, we use GenePix Pro, followed by the extraction, we use GenePix Pro 7. Now, yes, so in this particular project, which we are considering for COVID-19, and we look into the pattern of different proteins printed on the chip. So there, the steps associated with that, first of all, the scanning of the slide, then generate the image. This is called microarray image. So this is I think the one part of the image, because there are if you consider an image, will show briefly mostly in a live session also. You can see the whole chip. So there you can see different blocks. So this is the advantage of microarrays and for this work. So you can, for this chip, we are giving very importance in this project, you can assay multiple samples in a single chip. So it is obviously complex because of analysis wise, when we'll go ahead with the analysis. So one chip means one GPR file followed by different means subset of the data and process
one. So this is the first one. Now I'll go.
Audible?
Okay, suddenly we lost.
Hello.
Arka, are you there?
Yes, sir. Hello. Am I audible right now?
You're audible, but we can't see you or. All right, so till Arka gets set up and come back, idea here is that these slides what we are scanning has lot of information, right? You will see many signal, but how these signals are really good, that's something we need to bring out as compared to the background what we have, right? So that's what Arka is talking right now. The quality control on the microarray slides are important. Your slides are
Yeah.
seen.
Yes, sir. Yeah. So there is some technical glitches of this internet issues happened. So yeah, for the analysis of the data, first step is quality check of the microarray slides. As I already mentioned regarding the samples, so there are 14 samples we can process in a single chip. So there, intra block and as well as intra chip, that quality check we need to do as a finding out of the correlation coefficients. So there you can see the correlation coefficient must be closer to 1, that could be taken forward the data for the further analysis. So you can find that value is almost 0.9. So it's a closer to 1. So we can process the data in this way. So there also, you can also find out the outliers from that. So for that, the intra chip and intra block. So these are also could be considered the block as sub errors as well. So there we can process further with the correlation coefficient for the next step. So yeah, so now this is very, sir, I already mentioned regarding the it is a big data. So now if we focus a multiple number of chips, the output file is considered as GPR, that is called GenPix result file. Now we have developed our own algorithm. So here we can read the data the fastest, because the number you can see this is one part. So read chip number 5. So this is completely a complete GPR file with a single command in R, because we are writing the codes in R. So you can read the data. We fix a CSV file. So there we enlist the this particular four files, and finally we give a command. So they are different. We use the Lima packages for that, and they are read.ma images, this kind of libraries we use that, and finally process the data the fastest. So you can see this is might be 16 or 17 chips I am showing. So if we can see we can want to read 50 chips, we can do that because our algorithm is set in that way. Now after that, yeah, very important parameters is foreground intensities. In the foreground intensities, we need this one. You can see the chip 5, chip 6, chip 21. There you can see the number of these foreground intensities in this way. So next, after that, after this foreground intensities, next how you are eliminating the noise of the data. So foreground intensities for this one, yeah, so this is the foreground intensities of a single spot, and periphery considered to be the background. Now how we are considering the background, that is also a crucial point. Now segregating or eliminating the noise, for that we have used, sir also mentioned a very crucial point here, negative control probes. So how we are considering the negative control probes for finding out the signal to noise ratio, that could be the important part for the further data analysis. Now for the negative control, how you are finalizing the net, and it is completely based on your data, huge implementation of the coding expertise, because might be you're interested this negative control, as sir mentioned, the flag tag, let's say BSS buffers. But as per the SOP, you may require only BSA and buffer to be forwarded. But his tag or this next one, like sir said, the flag tag, that is also printed, but maybe we are not taking care for this analysis. So how you are eliminating this, because this could be taken as a negative control. But how you are eliminating specifically these two features, his tag, from this part, from this overall list, considered to be a vector for R, how we are considering or eliminating these two. This is also coding expertise. So I'm trying to just balance it, how a biologist and a data analyst is working together to finally come up with the for the result. So now for the SNR one, up to you're getting to the SNR, you need to find out the average. So average means the proteins which are printed, these are in duplicates, like this. These are in duplicates. So these are the duplicate spots. Now how you are finding out the average out of it? F1 plus F2 divided by 2. This is the logic. Now you need to read the data in this. So if we can consider two data frame in R, let's say for the spot 1 and spot 2, 1 and 1, just this plus this divided by 2. How you can proceed? Loop algorithm, just loop using for loop or while loop in R. So this way you can find out the number of features at a single time, single point, because this is the advantage of coding. Otherwise, you need to take every block and go ahead for this spreadsheet work, which will take lot of time because it is a big data. Now you just see next part is very crucial to find out the signal to noise ratio. So for signal to noise ratio, average spot intensity divided by the intensity 25% quantile of the negative control probe. So how a coder is going ahead? First, he or she needs to finalize the negative control probes, then finding out its intensities, which already we got from the table, and then divided by the 25%, 25% quantile you also need to find out. So then it to be divided or normalized with the specific one. Let's say we are taking one simple one. This is the list because this is the negative control. We are segregated in this way because we need to divide the actual probes intensity divided by this one. Now, so this is one distribution. So how a coder will go ahead. So this is the first one, feature specific normalizations, or considered to be the finding out the SNR block wise. You can see the medians are not uniform. It's obvious because we have not done any kind of global normalization techniques. So this is feature specific. So whenever we are considering, this is also normalization strategy, because you are considering one probe intensity, you are dividing it with the 25% quantile of the negative control probes. This is kind of a normalization, but this is not a global one. So now you can see it is not uniform. So next, the data analyst or data scientist should take a call which normalization can give me a uniform one of the medians, because we are now looking into the sample wise. Because till now, one sample, one block. Next, multiple samples. So for multiple samples, you need to go ahead with a different strategy of the normalization. Which could be the strategy? I'm not going into detail of that because a lot of strategies are there, but still for microarray and specifically for protein microarrays, we need to go ahead with the quantile normalization. Just looked into that. Now it's completely uniform. The medians are uniform. Now this is one strategy, but for peptide arrays, mostly my colleague will give you some suggestions, but detail about peptide arrays. They are completely taken a different call of going ahead with the G-score technique. But for protein arrays, we should go ahead with the quantile one. So next, there are different trajectory to showing the data. Data visualization is one of the major part for our projects. So we are using R coding for not only analysis, it's the one part, then substring. If we go ahead with this major part for the visualizations one, we go ahead with the R. So there are a lot of packages, but let me tell you one package which is very crucial for our analysis to show the best data, which is called ggplot. So now after this is one part, there our interest is to show a few features which is showing, let's say this spike one, how we are expressing or showing the data above 6 SNR. So 6 SNR is a value which is quite confident. We are confident on the data. If we set the SNR value more than 6. I'm not going much in detail because if you consider mu plus 3 sigma, it will provide you, if you consider a Gaussian plot, and if you just focus in mu plus 3 sigma, beyond that, your data should be very strong. So accordingly, if you consider the 6 ones of the SNR one, then we are already past the confidence interval. So that's why in this way, we just wanted to show the variations in different samples for a feature using this plot. So now next part here, this is another, now I'm going bit in a detail of the statistic, but not in much detail, glimpses. So after doing that, there are different strategy involved for the data analysis. What are those? We can't take a instant call that which statistics to be taken forward. In this case, why ANOVA? Because data is already that is already normalized. This is not a when we have not stopped there. further two another filter we have imposed, which we need to check the variance as well as another part normality test. So these two tests could be done. Then only we take the call. Either we'll go ahead with the ANOVA or not, because there are other strategies are there, which is called critical Wallis test. If your data is you are not okay with the normality, the researcher could take the call to go ahead with the critical Wallis as well. But here you can see we have go ahead with the ANOVA. Why? Because there is another rule. We are considering cohorts, multiple cohorts. 1, 2, 3. You can see these three cohorts we are looking. So when you are going for the multiple cohorts, you have, you need to take with the data for either ANOVA or critical Wallis. If the individual one, student test, man with new test. So these are the technique. So there is the multiple. So we go ahead with the ANOVA. So these are the basic of the few features. But as a data analyst, consider it as a feature. I do not. It might be SARS-CoV-2 and other proteins. But for us, when it should come to us, we should take the call. So this is the sample, this is the features. We should provide the information to our colleagues, our biology colleagues, so they will take the call, the how they will take it forward. So yeah, so this was, and this significance is very important. Yes, we have developed our indigenous code. So you can find it out, the star which you are imposing here. This is completely indigenous. We just need to find out the p-value from your this significance, this our analysis. It will automatically fix the star. So this way, actually, we proceed. So this is also in the, we are just recently, we have just completed the project. So we have given you the glimpses of that. And finally, most important part, whatever you are looking for the data science point of view, finally the validation. How you can do that? Using the raw data from the screen itself. We'll show briefly mostly for our system. So there you can find out the screen, how we are taking the block. If you are considering, so I've just shown you the pattern, one violin, another. So you can see the trend, but you mostly found the maximum intensity in this another one. This is called post-pandemic. So you can find it out here. This intensity actually came. So this way we also check the raw data, and so these are the outcomes. So thank you so much. So thank you, sir, and thank you everyone.
Well, thanks a lot, Arka. I think you have explained very well, and of course, you've done a lot of research in this area. So let me now invite Surbhi Bihani. She is another senior PhD student who is biologist, who has been looking at these arrays, what the significance of this data means, while Arka was on the technical side looking at the data analysis and the device part. Surbhi has been looking at how to make sense of this data in context of the immune responses for the patients who are suffering from COVID-19 or other infectious diseases. Surbhi, for next few minutes, over to you to show the applications of SARS-CoV-2 peptide arrays and how we have used them for some of the clinical applications.
Yes, so I'm just sharing my screen. I hope it's visible now.
Yes, Surbhi.
Yeah. So I'll be talking about how we have used peptide microarrays to interpret some important information about the immune response to SARS-CoV-2. So I'll just briefly again give you an overview of the experimental workflow. As the professor already mentioned, the peptide microarray is composed of overlapping peptides of the entire SARS-CoV-2 proteome. And we incubate those microarrays with the patient plasma, then incubate them with secondary antibody against the human antibodies, and then acquire image on a microarray scanner. And from the scanned image, we extract the data and we use that to interpret the immune response, whatever we are looking at, be it IgG, IgA, or IgM. In this case, we have analyzed IgG response and against all the proteins of SARS-CoV-2. So here you can see this is a color map showing the landscape of IgG response in COVID patients, and we have compared that to some of the naive patients. So what I'm showing on the slide is for few proteins like NSP3, NSP12, spike, nucleocapsid. And the blue dots that you're seeing is where we got the response. These are the patients. One is the naive cohort. These are the patients that were either negative for SARS-CoV-2 or few were for samples that were collected before the pandemic started. So they were absolutely not exposed to SARS-CoV-2. Then we had some severe patients, and then we also had a cohort where the patients were recovered. So the blue lines are where we got the signal, which corresponds to IgG reactivity against a particular peptide in this particular protein. So as you can see, even in naive, there is you'll see some response against few of the proteins of SARS-CoV-2, which means that there is some level of identity between the previously existing human coronaviruses and SARS-CoV-2. But you'll see that in spike protein, it's the least. So spike, we all know that SARS-CoV-2 is a novel virus, and the spike protein is what is responsible for the infection. So you will see least identity over here and high cross-reactivity in other non-structural proteins. Then these are the total number of peptides, the epitopes that we were able to identify for different number of proteins. So as you can see, NSP3 has a very high number of epitopes for the simple fact that it's a very big protein, first of all. And secondly, this protein is responsible for some of the crucial viral life cycle processes. So this is most likely to be conserved across the coronavirus family. Similarly, NSP12 and NSP12 also showed a high number of immunoreactive epitopes. And since we also had a naive cohort, we were able to look at some cross-reactive epitopes. That is, even in naive patients, we were able to find IgG that were reacting with the SARS-CoV-2 peptides. These are the number of epitopes for spike protein, and as you can see, it has very less number of cross-reactive epitopes, which tells about the novel status of the SARS-CoV-2. Then we were able to map the identified epitopes on the 3D structure of the spike protein, so that we can analyze the location, like where exactly that epitope is and how that is related to the molecular function of the spike protein, like from where the spike protein is binding to its receptor, and whether the epitope against which we have seen reactivity is actually involved in this interaction or in any other function of the protein. So we were able to locate the epitope on the 3D structure of the spike protein. Similarly, we also did that for nucleic acid and non-structural protein, as non-structural protein 12. Now, since we had two cohorts, one was severe and one was recovered, we tried to look at whether some of the epitopes were associated with recovery or severity, and we were able to find that some of the peptides, they were mostly reactive in recovered cohort. So we hypothesized that these antibody response against these epitopes, so these peptides, are associated with recovery. So one of them was this particular peptide in the spike region, which was highly associated with recovery. And it is here at the junction of the S1 and S2 subunit. And this is where this region actually facilitates the entry of the virus into the cell. And finally, the microarray slides. On the microarray slides, we also had peptides with mutations incorporated in the peptides for the common SARS-CoV-2 variants. So you can see these are some of the mutations for Delta, Omicron, Gamma, etc. And so what we saw was for the same region, but when there was a mutation, the antibody reactivity was different. So what we tried to look was for which mutation which mutation was responsible for decreased or increased antibody reactivity. So you can see for some mutations, the response in the wild type peptide is more. For example, this particular mutation in the E484K in beta and gamma, the response is higher in the wild type and lesser in the mutant peptide, which means that this mutation is most likely responsible for decreasing immune reactivity. Whereas there were few mutations like this one G142D in Delta and this one in Omicron, where the response was more in the mutant peptide, which means that these mutations are most likely responsible for increasing the immune recognition. So these are few of the applications of how we have utilized peptide microarray to understand the immune response to SARS-CoV-2, and these can be applied to various other diseases as well. So that's it from my side. Thank you.
All right, thank you, Surbi. So now I request Surbi and Arka to just give a glimpse of some of the microarray setup and the scanner, the slides, what we have in the lab. Although we have shown some, experiments and overview, but it will be good for you to show from lab directly.
Yes, sir. Thank you so much. So all of you have just heard about this, about our data analysis and the experiments, the protocol. So now let me show you about the scanner and how we access the data or how we extract the data. Yes. So this is our scanner. So this in this place you need to insert the chip, and we are actually having some time issues. So now we are not scanning the slide. So directly I'll show you this images. So yeah, so this is the protein array image. I think you can see, as I mentioned. Now I will go, or I will tell you about the duplicate ones. Yeah, so these are the duplicate spots. This. So these are printed in duplicates. Now I already told about the QC steps. Now next call is to next step is definitely gal file. So now I'll directly go to the this one. So this is the gal file. Gal file is called as GenePix array list. It is protein, this annotation of the protein or peptides. But this grid automatically will get. But we have also recently developed our indigenous gal code that we can generate the gal file the first test. So this one, this is the gal file. The circular grid you can see. This actually helps you to segregate the foreground and backgrounds. So now after that, how we extract the data? There is another. This is called the GPR one. So for that, you just need to click on this. This is the data. If we just click here, you can see the data is generated. This is the data. Now after that, you need to just save the data as save results. Here you can see the extension .gpr. So this is enough. So after that, but for that, I have shown you this directly, this one, but you need to do a lot of things like yeah, so there are the block boards. So initially when you place the grid, it is not uniform. You need to place it properly. Now it is protein array. Now let me give you just a brief about the peptide arrays, which should be said. So this is the peptide arrays. You can see this one. So this is the peptide array. So I think if this is fine. Yes. These are the peptide arrays, and extraction of the data remain the same, but here we are not using GenPix Pro. Here we use Mapix for extraction of the data.
All right.
Okay, so this is all about. Thank you, sir. Yeah.
Maybe we could show some slides.
This slide.
What slide?
So we could show some microarray slides and
Yes, sir.
Okay.
So Surabhi is arranging that. So let me tell you something about this is actually Surabhi said about the peptides, and this is also computational where now if we just find out about the epitopes that also Surabhi mentioned. So there is a difference between peptides and epitope. Epitope is the chain of the peptides, contiguous peptides. Yeah, so this is the epitope. So now let us show you this chip. So this is the microarray chip. This is the peptide arrays. Yeah, this need to be just open this one and then.
Right. So again, like, it highlights that on a very small glass slides, which students use for different experiments, one could actually have the thousands of spot or the peptides or protein, which in a very miniaturized way represent the entire proteome. And that could be screened with the high density arrays. All right. Thank you, guys.
Yes, sir. Okay.
Let me move on to the next application, which is essentially thinking about how to use the microarray for the cancer-related application. What we just saw was the SARS-CoV-2, right? But again, same type of platform we also use for the cancer screening, cancer autoantibody biomarker screening. And here we use the human proteome arrays, where we have the almost 19,000 human protein, which is with duplicate spots, control spots. And then we have a screen for different type of brain tumor patient, where we look at the control serum sample on the lower grade and the higher grade of the patient, and trying to look at is there any pattern of antibody which is different from these individuals, right? So this could be again utilized for the even cancer autoantibody screening, not only just the SARS-CoV-2. Additionally, in addition to the protein microarray, which is the fluorescent-based signaling, what we are looking at the readout, there is a school of thought that why not we look at intrinsic properties of molecule and try to take two molecules and see when they interact, what kind of changes happen when the two molecules comes into the interaction, whether mass changes, whether the dielectric constant gets changes, or the refractive index of the medium gets changed, right? So based on that, many type of label-free biosensors have been devised. We have been also doing some work during my postdoc to look at how the microarray platform could be coupled with the label-free biosensors to provide the high throughput screening for various type of biological problems. So in this case, there are different platforms, different principles involved, like carbon nanotubes or the planar wave guide arrays, nanowire sensor arrays, spectral reflectance imaging biosensors, nanohole arrays. But one which gets more prominence is surface plasmon resonance, which is quite being utilized as a good platform for looking at the protein-protein and protein-drug interactions. More recently, another platform which has also been quite utilized is known as the BLI, which is based on the principle of biolayer interferometry. Nirja from our lab is going to talk briefly about this part.
The technique of biolayer interferometry is based on the analysis of the interference pattern of the incident white light, which reflects from two surfaces. First, the immobilized molecules of the biosensitive, and a reference layer, which is nothing but the sensitive without the immobilization. This technique has a plethora of applications. Major purpose of it is to determine the binding kinetics of two molecules at question, which describes the dynamic binding interaction between those two molecules. These interactions are typically expressed as K subscript small a or K small a, the rate of association, and K subscript small d or K small d, the rate of dissociation. The ratio of these two gives the equilibrium dissociation constant or affinity, denoted by K subscript capital D or simply KD. It can be also used to determine steady state constant. The steady state is a term used frequently when describing enzymatic processes. It is a situation in which the rate of formation is balanced with the rate of destruction. It is also widely used in antibody-antigen interactions or any protein-protein interactions for that matter, along with antibody affinity rankings. It is also used in small molecule-based drug discovery, which is to determine the interaction and binding capability of a proposed drug molecule to the target protein of interest. Absolute or relative sample quantification can also be performed using this technique. Epitope binning is another interesting application of it, which enables the differential analysis to discriminate between antibodies that bind the same antigen, but not the same epitope of the antigen. In a nutshell, almost any interaction-based studies can be planned to be performed using this technique. This is an exemplative study performed on assessing the binding of the receptor binding domain, RBD, of the SARS-CoV-2 spike protein with the ACE2, the angiotensin converting enzyme 2 receptors. On the left, we can see the experimental workflow. They have shown the different steps of it, of the antigen loading first, then the subsequent antibody bindings, the washings of the non-binded, and finally the detection. One thing there is to note, after every binding step, there is a shift in the wavelength. See, so this shift in the wavelength of the returning light from the biosensor tip is from the interference of the biosensor surface, and this is what is registered as the interactions. This is a representative of interactions, the wavelength shifts. That is, how it is quantified. On the right, you can see it is a representative result image showing the significantly increased interaction, denoted by the obviously increased shift in wavelength, as I just told, of the seropositive individuals, denoted as SP, or which are basically the recovered patients, having the antibody of the antigen here we are talking about, when compared to the seronegative individuals, SN. So this is one of the exemplative studies.
All right, so bilayer interferometry is one of the platform, and additionally, the other platform which is of quite interest is SPR, surface plasmon resonance. How this works, let me just share my slides and quickly give you an idea. Of course, in this course, you have been studying this also in quite detail. Idea is to look at how the refractive index of the medium gets changed when the two molecules come in contact with each other. And for that, we are using the prism, gold slide, and looking at how the sensorogram changes in response to the binding. And based on that, the on rate, off rate, and the KD values are being calculated, right? So SPR, we have covered quite a bit in the course. I thought to show you again live how the this kind of experiment can be performed using SPR. So let's just have some lab view of the SPR workflow.
Sensor chips, biosensor chips, on which we immobilize one of the interacting partner and we pass the other partner. The partner which we immobilize is called as ligand, and the other which we pass is called as analyte. So in DRP PT100, there is a chip compartment where we drop the chip. Okay, you put your chip here, and this is sample compartment. We put your sample here, and this is buffer compartment, and this is discard. So you immobilize your one of the interacting partner, and then you will pass the other partner, and you will get this association and the dissociation. So for immobilization, you use two types of strategies. One is capture, and other is direct. Either you can directly immobilize one of the ligand, or you capture. For example, in direct immobilization, you use different types of chemistries like amine coupling, thiol coupling, or aldehyde. So, for example, in amine coupling, your amine group of your protein is involved. So, this chip has a carboxy mechanism. You activate that group, and this activated carboxy methylated group will bind to the pre-amine of the protein, and your protein will get directly immobilized. For example, in capsule method, you have nickel NTU on your chip, for example, or a small nickel NTU with so this type protein will get captured on the nickel NTU. So this is how you immobilize your one of the interacting partner. Then you will pass the other interacting partner. Then you will get the association. Okay. After that, buffer will pass. This will give you dissociation. The ratio of this association rate constant and the dissociation rate constant will give you dissociation. Will give you equilibrium dissociation constant. That is the strength of your interaction. So after passing of your analyte, if the analyte is not getting completely dissociable, you will need to regenerate that. For that, you have to use different regeneration buffers, and you have to optimize that. So after regeneration, you will get the sensogram like this. So for example, this is the association, this is the dissociation phase, and here you regenerate. Okay. So you have different types of chips are available. On basis of what experiment you want to perform, you will choose the chip. For example, if you want to perform, if you want to immobilize P-stack protein, you will use LTA. If you want to immobilize lipid, you will use L1. If you want to immobilize biotin nucleic acid, you will use SHA. This is how, on basis of your experiment, you will use different types of chips. Now, what you get? So when you pass your analyte, you will get the association. This is how the association looks, and then buffer will pass, you will get the dissociation. So this will give you the bigger. This will give you a small Ka, the association rate constant. This will give you the small Kd, that's a dissociation rate constant, and its ratio will give you bigger Kd. That is dissociation, that is equivalent dissociation constant. That means this will give you the strength of interaction. More is the strength, smaller is the Kd value. Okay. So why do we use SPR? Like what is SPR? What extra information SPR is giving? So you can get Kd value from other assays also. Like for example, like ELISA. For example, any other instrument, you can get the Kd. And why SPR? So if you can see here you can see there are different types of sensogram. Okay. So you can see the shape of the sensogram. This. So this is in this example, this five have a same Kd. Kd is the same, but this association constant and the dissociation constant is different. For example, if you see here, the association is regular example in this green sensogram. You can see the association is very fast, but the dissociation is very slow. Here in blue, the association is optimum, dissociation is slow, but as compared to green, it is still fast. So this information will reveal a lot of information. You have a same Kd, but different small p and small Kd. That is, this will give you K and Kd.
All right. Well, so thank you, Rusha, for the SPR. But the time is running short, so we will leave this particular session right now. What I'd just like to conclude here to the students that this whole field of interactomics and multi-omics, right? It's going to open up a lot of opportunities for you in future. If you can think about these technologies as a good way of generating your, biological information, clinical information, and utilize that towards building the hypothesis, building the models, and just don't be so biased. Look at the data in much more comprehensive manner. Think about layers of information in the systems approach, how you are going to utilize them together, and then trying to understand the system better. So the researchers, the students who are trained in this area of looking at the data with more holistic approaches are going to be in the forefront in the, driving the innovations in the upcoming years. And I think all of you who are being part of these courses will be very well placed for taking this lead and these initiatives in future. I hope the courses of MOOC, especially the Interactomics course, has been helpful for you to give you the new perspective of these technologies, how you can utilize them for the biological applications, the biopharma applications, and of course, towards your future career development and your academic journey. With this, let me thank all our TAs who have been involved in this course, especially Amrita is there, Ankit, Amit, and today we have also had the special request to Surbi and Arka. And all of these TAs have together tried to work with you to address your queries on the forum, looking at the live sessions, as well as giving the live activities from the lab to give you the much better feel of how you can do this experiment yourself. I hope the course has been enjoyable for you as much as we enjoyed interacting with you and teaching this course to you. Wish you all the best for your exam and for your future academic success. Thank you very much.